Hello everyone, and welcome to this audio podcast. As part of the Contemplate. This audio podcast is part of the Labostix, Blizzard, and the specials. This audio podcast is part of an Echo and Update. This audio podcast as part of a Storm Labostix production. Through this audio podcast and looking at some of the well-drafted works of M. Bohtha, and where the student had looked at sort of diagnosis methodologies utilized to determine SARS-CoV-2 or COVID-19. This audio podcast will look to go into some of the molecular bases, some aspects of the molecular bases. So C1q, a serum protein, will bind to antibodies on pathogens and activate the sort of classical immunological pathway of the complement system. After several steps, the membrane attack complex is formed on the surface of red blood cells, creating or then prompting sort of holes on the holes within or on the membrane. As part of the analysis, as a basis, therefore hemoglobin is then released, and it is measured or determined by spectrophotometry, and there is a part of the spectrophotometry which is referred to as a CH50 point, which is where a specimen at that point it allows investigators to determine the sort of the amount of hemolysis in around the effectiveness of SARS-CoV-2 or COVID-19 in amongst specimens at quantifiable on a quantifiable basis up to at 50%, and which is found, which then can be which is where in amongst the hemolysis at 50%, it can help laboratory investigators to determine sort of the amount of activity whereby the complement system of the immune system from the from the immune system, which reacts as part of the classical pathway, and this is identified, and this can be identified in amongst the analysis that investigators would have done from patient X's blood sample. An investigation, a sort of investigation referred to as a Western blot, which falls within the basis of a molecular investigation, sort of early molecular investigations, which had been done to identify the amount of expression and estimate of in around complement C4 and complement C4 alpha and complement and C4 alpha proteins from a mixture, and to do the Western blot investigations. First, one of the main bases is that it has to be where there's a separation of particular proteins by the use of gel electrophoresis. And then there is a sort of a transfer of the protein to from the gel to solid support, and finally detect by using target proteins. This is then a way for lab investigators to determine sort of the basis in around the sort of molecular aspects of SARS-CoV-2, for example, and then it is where for example the C4 alpha is so in this particular works in amongst patient X, it was where C4 alpha was overexpressed compared to healthy human serum, which was used as a control. And the C4 levels, or C4 alpha levels, have been similar, have been similar, but at the same time, the molecular basis was higher. The molecular basis of this was higher in the case of patient X. So after the transfer, in as part of the molecular, in around the Western blot investigation, blocking agents could be used to reduce non-specific binding surfaces. The primary antibodies, which are C4 alpha antibodies, are ways wherein there is a bond between membrane and protein. The secondary antibody is bonded to a primary antibody, and an enzyme which is bound to the secondary antibody cleaves the substrate, and cleaves the substrate, and this was used in another investigation, which referred to as chemiluminescence, which the substrate turns into light, and the product is blotted on a particular sort of film base, and in the patient sample, both C4 alpha and C4 both C4 alpha and both C4 alpha and sort of the molecular bases, the molecular bases of this were present, and in the classical pathway of the immune system, C1 cleaves to C4, and which then goes into C4a and C4b. C4b then goes into C2b, which forms C3. C3, which convertase, which leads to the formation of which then forms the membrane attack complex. And so, at the same time, it is through the use of spectrophotometry and the fluorescence emission, excuse me, the fluorescence emission. This allowed the investigators in around this works to determine the again, to determine the basis in around the virulence, but also in around the molecular basis of SARS-CoV-2, SARS-CoV-2, and additionally sort of and additionally the extent where that patient X had SARS-CoV-2 spread had spread within their sort of pathophysiology. So that draws this audio podcast to a close. This audio podcast is part of the Contemplate. This audio, excuse me, this audio podcast is part of a Labostix Blizzard in the specials. This audio podcast is part of an Echo and Update, and this audio podcast is part of a Storm Labostix production.